Good afternoon, everyone, and welcome to the Replace Educational webinars. So my name is Maud, and I work on the Replace project. So this project aims to collect the existing expertise on new approach methodologies, so alternative methods to animal testing in one central open access database. And we are organizing this series of educational webinar with the aim to put the expertise we have in the database in the spotlight. And the webinar of today will focus on 3D models and organoids. So today we will have two presentations. So each presentation will be about 20 minutes, and then we will have the question after each presentation. So if you have any questions, you can put them in the chat and we will answer them after each presentation. So this session is also recorded and will be placed on the Replace YouTube channel in the weeks following the webinar. And this webinar is also accredited by the Flemish and Brussels regions. So if you would like to receive a certificate of attendance, you can put your name and email address in the chat during the webinar. And I will provide these certificates later on. So we will now start with the presentations, and I will introduce the first speaker of today. Cyril Corbet is a Principal Investigator, FNRS Research Associate at the UCLouvain in Brussels. He started a PhD in biomedical sciences at the University of Lille in 2012, and then moved to Belgium to start a postdoctoral research stay at the group of Professor Olivier Ferron. There, he gained a strong expertise in the study of tumor microenvironments and how microenvironment peculiarities result from, but also drive metabolic preferences in tumor cells. His work was published in several high-impact journals and was also recognized by different awards. His current research group, created 4 years ago, is now working on the interplay between tumor microenvironments and metabolism in the context of therapy resistance. So the floor is yours.
Well, thank you very much, Maud, for this for the invitation, also for this kind of introduction. I will now share my screen. I guess it's fine. I hope everyone can see my slides. So again, thank you very much. So today I would like to share with you in the next 20 minutes what we are currently doing in my group about the study of tumor metabolism and how we can use 3D models like tumor spheroids or tumor organoids to better understand intratumor metabolic heterogeneity and what are the current limitations as well of the use of these models to study tumor metabolism. So this is the outline of my talk. So I will first give you some insights about what is tumor metabolism and what is the challenge when we want to study tumor metabolism in different contexts. And then I will share with you some studies that we already published in my group in the last 5 years to study metabolic heterogeneity according to the change of macroenvironmental conditions by using different 3D culture models, especially spheroids at the beginning. And now we moved more recently with the use of patient-derived tumor organoids. And at the end, I will share with you what are the current limitations and challenges for these models in order to initiate the discussion as well with you. So when we talk about cancer metabolism, of course, it's important first to start with what is the fate of a cancer cell. A cancer cell have 3 main objectives, which are to survive, proliferate, and migrate in order to support disease progression. And how these cancer cells can do this is by producing energy like ATP in order to support all these phenotypic changes, but also by producing building blocks in order to produce two cells from one cell. And obviously, this energy and building blocks will come from cellular metabolism, and especially with a variety and a complexity of metabolic pathways occurring within the cells, which allow these cells to produce energy and building blocks according to the use of different substrates like carbohydrates, amino acids, and also fatty acids. So when we talk about tumor metabolism, it's a complex situation because we can easily understand that these metabolic preferences will change in a dynamic state according to the stress conditions that the cancer cells have to face. When we talk about some stress condition, we talk about, of course, some treatment. When the tumor is treated in the clinics, the cells will be stressed and they will have to adapt their metabolic preferences in order to survive under these treatment conditions. But it's important also to understand that the metabolic preferences will be really different when the cells are in the primary tumor or when they are circulating in the bloodstream or when they reach secondary organs in order to metastasize. And even more importantly, this leads to two main contexts and concepts, which are the intratumor metabolic heterogeneity here, which is mostly the influence of the microenvironment, I mean hypoxia, acidosis, absence or presence of nutrients, but also the cooperation of cancer cells with a variety of different cell types within the microenvironment, like immune cells, fibroblasts, that will change also the metabolic phenotype of these cancer cells. And obviously, a second concept is the inter-organ metabolic heterogeneity. We can understand that the microenvironment, the local microenvironment of cancer cells will be very different, whether we are in the brain or in the lung or in the liver, for example. And this leads to two different metabolic scenarios that can occur in tumor cells in order to adapt their metabolism. The first concept is what we call metabolic flexibility, which is the capacity of cancer cells to use different substrates in order to produce energy and building blocks. The second concept is what we call metabolic plasticity, which is the capacity of these cancer cells to use the same substrate, like glucose, for instance, but through different intracellular pathways. For example, if we take glucose as an example, cancer cells can use it through glycolysis or through pentose phosphate pathway, for instance. And there are again other scenarios like metabolic symbiosis, which is a cooperation between cancer cells with other cell types, which can be other cancer cells located in another region of the tumor, or other cell types like fibroblasts or adipocytes that will provide substrates for the cancer cells in order to use them as bioenergetic substrates. Of course, behind this cooperation, there is also competition for different substrates, and cancer cells can compete, for example, with immune cells for the same substrate in order to produce energy, but it's also a way to inactivate the immune system by shutting down the availability for this substrate. And finally, there is also the direct transfer of mitochondria or mitochondrial DNA between different subtypes like stromal cells and cancer cells, again, in order to find new strategies to produce energy in these cancer cells. So altogether, it's easy to understand that when we are culturing our cells, as many of us are doing in our lab with cell monolayers, with medium in our incubator, we are very far from the this in vivo situation. Again, most of us are culturing our cells in incubators with 21% of oxygen in a medium which is buffered at pH 7.4, and we are always replacing the medium every 2-3 days in order to provide cancer cells with all the energetic substrate that they need. And obviously, all these conditions are the same for all the cells within the plate. So again, we are very far from what I just explained about this metabolic heterogeneity. And this is why more and more we are now using 3D models like spheroids or organoids in order to better mimic the in vivo situation by the creation and the occurrence of gradients within these 3D models, gradients for oxygen, nutrients, growth factors, which will be less and less available more you are going deeper within these 3D structures. And at the opposite, the washout of metabolic waste like CO2, protons, lactate will be more difficult in the core of this spheroid. So there will be an accumulation of this waste in the core of these 3D models, creating a gradient from the periphery to the core of these 3D models. So in the last 10 years in my lab, we have been especially interested in one hallmark of the tumor microenvironment, which is acidosis, which is the accumulation of protons in the microenvironment and which comes from a dysregulated metabolism in cancer cells. And most often this acidosis is commonly associated with hypoxia, but it's even more complicated. So here we created this model where there are cancer cells which are close to the blood vessels. They have access to oxygen and they can use an oxidative metabolism. But more you go deeper in and far from these blood vessels, and cells will have to face some hypoxic conditions. And here they will turn towards anaerobic metabolism. But there is an intermediate compartment where oxygen is still present and with a variety of substrate. And this compartment is mostly acidic and moderately hypoxic. And in the last 10 years, we try to understand how this acidic compartment could influence the metabolic preferences in cancer cells. So again, it's a very old paper reporting that acidosis and hypoxia are not overlapping in tumors. So here these are measurements of extracellular pH and PO2 in tumors directly, and we can clearly see that the gradients are completely different within the tumors. So there is no perfect overlap between acidosis and hypoxia. And this is something that we can also assess in our different in vitro or in vivo models by using what we call the flip peptide, which is a pH-sensitive peptide that allows, when it's conjugated to a fluorophore, to discriminate and to image the cancer cells which are exposed to acidic conditions. Indeed, when the extracellular pH will drop, there will be integration of this peptide within the cell membrane. And we can detect this peptide through fluorescence. And this is what we did on tumor sections. Here you can see on colorectal cancer tumor, the red signal, which is the flip signal, meaning the acidic regions, and on the right, pimonidazole, which is a commonly accepted marker of hypoxia. And you can appreciate that there is no overlap or no perfect overlap between acidosis and hypoxia, confirming that probably these two hallmarks of the microenvironment can certainly differently influence the metabolic phenotype of cancer cells. And this is what we continue to work on by using 3D spheroids. Here, again, by using the flip, we could see in our 3D spheroid that at day 1, there is almost no red signal for the flip, so almost no acidosis in these 3D models. But more you wait for the culture, bigger are your spheroids, and you can start to see a red signal occurring after day 5, day 7, which is the natural occurrence of this acidic core in spheroids. And we can play with this, and we can compete with this acidification if we incubate the 3D spheroids with an excess of bicarbonate to buffer this acidosis. We lose the red signal within these cultures. And by using them, we could document that the cells which are exposed to acidosis, so the red spots, are also the ones that accumulate lipid droplets by modifying the fatty acid metabolism in these cancer cells. And this is something that we published already some years ago. And as a follow-up study, we decided to investigate whether we could take advantage of this fatty acid addiction in the cancer cells which are exposed to acidosis. And to do so, we tested some dietary approaches by providing exogenous fatty acids to cancer cells, either saturated fatty acids that you can find in cheese, in Nutella, for instance, monounsaturated fatty acids like oleic acid, and also a variety of polyunsaturated fatty acids like omega-3 and omega-6 polyunsaturated families. And interestingly, again, by using 3D models, we found that saturated and monounsaturated fatty acid had almost no impact or no detrimental impact on the growth of 3D spheroids, as you can appreciate here. But when we were adding an excess of polyunsaturated fatty acids in the culture medium, you can clearly see that it induces a cytotoxic effect in our 3D models. And again, here by using PI staining in order to image apoptotic cancer cells, I hope you can see that this omega-6 and omega-3 treatment mostly induced apoptosis in the core of the spheroids where the cancer cells are exposed to acidosis. So back to this situation when cancer cells can use a variety of different scenarios to adapt their metabolic preferences. It highlights the complexity of this metabolic network, and obviously this highlights the complexity of targeting this metabolic phenotype. Indeed, if we come with a metabolic drug, the cancer cells through this network, this highly complex network of metabolic pathways, can easily find a way to overcome the inhibition of one pathway and to adapt and resist this treatment. But in a recent study, we tried another approach, which is by using a metabolic drug. It was to force the cells to adapt and to be dependent on metabolic pathways that could be targeted in the second way. This is what we call collateral lethality. And again, we use as an example 3-bromopyruvate, which is a metabolic drug which will preferentially inhibit the glycolysis pathway. And we found that when the cancer cells become resistant to 3-bromopyruvate, I will stop here, and that when we incubate resistant cancer cells and native cancer cells, which are sensitive to 3-bromopyruvate, we have a specific organization of these two cancer cell population within our 3D models, with the green cells, which are the GFP expressing cells, so the native cells, being located at the periphery of the 3D models, while the resistant cells are more exposed, are more located in the core of the 3D spheroids. And this is associated with a specific metabolic phenotype and the dependence towards glycolysis that we can target with diclofenac, which is repurposed as MCT4 inhibitor. And we can see that when we come with diclofenac treatment, specifically on resistant cancer cells, they become more sensitive to this treatment in comparison to the native cancer cells. And again, we can see by propylium iodide staining that this treatment is mostly effective in the cells located in the core of these spheroids. The second strategy was to use a metabolic inhibitor to improve response to conventional treatments like radiotherapy. And here we use 7-ACC2, which is an inhibitor of MPC, which is a transporter of pyruvate towards mitochondria, in order to force and to reoxygenate the tumors. And here we can see that this 7-ACC2 has already some dramatic cytotoxic effect on 3D spheroids, but more importantly, by inducing a reoxygenation of the tumors, there is an improvement of radiotherapy application on tumors, but also on spheroids. So finally, in the last few minutes of my talk, I would like to switch to a more recent study, and what we are currently busy with in my lab. It's the use of patient-derived tumor organoids, which are closer to the clinical situation than 3D spheroids, which are established from cell lines. So as you all know, probably, tumor organoids are kind of mini tumors or tumor twins, since they are like a tumor ID card, since they recapitulate the genetic profile of the tumor, but also the therapeutic response. And here is just the workflow of what we are doing in my lab for breast cancer organoid models, for instance. And on this, in this study, what we're interested in is to see whether we can also add the metabolic profiling of these organoids in order to add a new layer of sophistication for this study. And in a recent study, we mostly focus on triple-negative breast cancers in order to try to understand how metabolism was reprogrammed when cancer cells were exposed to neoadjuvant chemotherapy, and also can we predict the occurrence of resistance to neoadjuvant chemotherapy according to the metabolic profile. And for this, we use different organoid models, all of them being established from treatment-naive patients. And we use a variety of different metabolic approaches in order to profile these organoids. And to cut a long story short, what we found that when organoid models are exposed to chemotherapy, especially paclitaxel, we have an increase of glycolysis with increase of glucose consumption, lactate secretion. And when we come with a glycolysis inhibitor, we can clearly resensitize and improve the response to chemotherapy in comparison to single treatments. So there is really a potential to take advantage of this metabolic reprogramming to improve response to chemotherapy. And finally, we found that in tumors and in patients, this is the same situation, since if we compare the transcriptomic profile between responders and non-responders to chemotherapy, we found that glycolysis is one of the top upregulated hallmarks in the non-responders, as we could find in organoid models. So all of them is also a way to validate the findings that we had in organoid models. So as take-home messages, since I'm almost at the end of my time, I hope I could convince you that 3D tumor cell cultures are key to recapitulate first gradients that we can find in tumors like pH, oxygen, and nutrients to better evaluate and assess metabolic heterogeneity, that these 3D models are relevant to test metabolic drugs and to see whether we can improve response to conventional treatments. And more recently, that metabolic studies in patient-derived organoids could be the next step in order to develop new modes of personalized medicine. Obviously, there are some limitations for now in our model, but more generally in many studies, it's the lack of heterotypic culture systems, either in spheroids or in organoids. So the lack of cancer-associated fibroblasts, for instance, immune cells, adipocytes, in order to study the metabolic cooperation between these cell types. And here, just to mention a few protocols already described to generate these co-cultures in patient-derived organoids. And finally, there is still a need for techniques to better appreciate the metabolic heterogeneity within these 3D models. And here, just to mention what is now used as lifetime imaging microscopy to assess this metabolic heterogeneity. So with this, I'm done. I would like to thank all the members of my lab, of course, who did this work, all our collaborators, and of course, all our funding supports for this study. Thank you very much.
Thank you, Cyril, for the nice presentation. I see there is one question in the chat, but I have first a question for you. We have seen also in the field of cancer that there is also a lot now of targeted therapy. Have you also planned to study the impact of targeted therapy on the change in metabolism in your spheroid or organoid models?
Yeah, we have two different projects currently in my lab about anti-GFR targeted therapy, either in colorectal cancers or in head and neck cancers. And obviously, the idea is to find what is the metabolic reprogramming of the cancer cells which can survive after the treatment with such targeted therapy. So it's currently ongoing in my group.
Okay, thank you. So I will now read the question in the chat. So do you see a high variability within experiments using spheroids and organoids when you do the treatments, and how they respond in each replicate, for example?
Well, I would say that heterogeneity is certainly true for organoids, but I think that everyone working with organoids can appreciate this. For spheroids, it's I would say there is not too much variability when we treat our spheroid cultures with metabolic drugs or conventional treatments. It's more homogeneous in terms of response.
Okay, thank you. Second question is, are you looking at invasion or metastasis of collagen-embedded spheroids?
Well, this is something that we tried with spheroids. I would say that we were not completely convinced about what we could generate for this invasion assay. So it's something now on the side. Certainly, we will come back to this a bit later. But yes, I think it's important also in the way that we can use these 3D models to better study invasion or part of the metastatic process.
Okay, thank you. I don't see any other question in the chat, but I assume if other people have questions, they can also contact you. But for now, we will now move on to the second presentation. So I will introduce the speaker. So Amy Gloria Toulehohoun is a PhD student at the IREC Institute at UCLouvain. In the past 2 years, she has been working on gut barrier dysfunction in alcohol use disorder by focusing mainly on the intestinal epithelium alteration. To conduct this research project, she is using intestinal organoids to address specific questions associated to the intestinal barrier dysfunction, and the main purpose of using this 3D model is to overcome animal models' limitation in the designing and understanding of alcohol use disorder. So the final goal would be to modulate this model in order to correct the intestinal epithelium alteration related to alcohol use disorder. But she will tell you more about this just now.
Thank you, Maud. Can you see my screen?
Yes.
Okay. Okay, hello everyone. First, I want to thank the organizer of this webinar for giving me the opportunity to present my work. So, as Maud said, I'm second-year PhD student in gastroenterology lab of IREC Institute in UCLouvain. And our group work mainly on alcohol liver disease. And this group is led by Professor Peter Starker. So today I'm going to talk about the use of intestinal organoid as a model for small intestinal epithelium alterations in alcohol use disorder. So this presentation is structured as follows. I will start with an introduction and the definition of alcohol use disorder. Then I will talk about our previous result in the lab. And then finally, I will talk about the intestinal organoids. So alcohol use disorder is defined by the National Institute of Alcohol Abuse and Alcoholism as a medical condition characterized by an impaired ability to stop or control alcohol use despite adverse social and health consequences. So this condition leads to 48% related death in USA and 60 to 80% in Europe. Among the people consuming abusively alcohol, 90 to 95% will develop steatosis, which is fast accumulation in the liver. And a minority, 10 to 20%, will progress to more advanced stages, such as fibrosis and cirrhosis, leading to liver failure. Unfortunately, there is no FDA-approved treatment for any stage of alcohol liver disease, and abstinence is considered the most effective treatment. So why am I talking about the liver? It's because chronic alcohol exposure leads to bacterial overgrowth in the gut or in the intestine, and also it induces the loss of the tight junction between the epithelial cells. So as a consequence, we have the migration of bacterial or bacterial-derived products from the gut to the liver through the portal circulation. And in the liver, this will trigger pro-inflammatory responses, the increase of oxidative stress, and also the increase of collagen production leading to liver failure. But I will explain this more in detail way in the next slides. So in a healthy condition, the gut barrier is composed of different layers. So we have the defense barrier, and then that are the mucus that you can see here on the top of the epithelium. Then we have also the epithelium barrier composed of epithelial cells, and then we have the lamina propria composed with immune cells. However, in alcohol use disorder, we have what we call the gut barrier dysfunction. So this is associated with increased mucus production. Also, we have the reduction of immune response and bacterial dysbiosis. So as I mentioned earlier, this condition will lead to bacterial overgrowth and migration from the gut to the liver through the portal circulation. So before I continue, I would like to give you a description of the complex organization of the intestinal epithelium. So the intestinal epithelium is organized in two main compartments. At the bottom, we have the crypt, and at the top, we have the villi. So this epithelium is constantly self-renewing every 3 to 5 days to maintain the tissue homeostasis. To renew, we have the proliferation of the stem cells that are in the red here at the crypt base, and those cells will generate the ductal cells that will migrate here in the trans-amplifying zone, and these cells will continue their proliferation and start the differentiation, and once they are differentiated, they will migrate at the top of the villi. But only the Paneth cells will migrate back here at the top of the villi and stay next to the stem cells. And each one of these cells have a really specific functions. So if we take the enteroid side here, they represent the most population of the cells, and they are mainly involved in the absorption. But if we take the Paneth cells, they are involved in antimicrobial peptides and the defense in the epithelium. We also have the goblet cells that produce mucus in this epithelium. So previous works that are done by Luca Maccioni, which was the previous PhD student, he's now a postdoctoral fellow at the National Institute of Alcohol Abuse and Alcoholism. So his data show that in the epithelium of alcohol use disorder patient, there are some alterations. So you can see here on the left an immunohistochemistry staining of KIC7, which is a proliferation marker. So as you can see, we have an increased cell proliferation in the trans-amplifying zone of AUD patient compared to the control. But in the contrary, when you see here on the right, the base of the crypts lack proliferating cells. He also saw that there are some morphological alterations. For instance, we have the reduction of the villi in AUD patients compared to the control. And on the top of that, we also have an increased number of goblet cells. So these results were only observation made on the biopsy of AUD patients. So we were looking for a model to establish a direct cause-effect relationship between alcohol and this alteration, because we don't know if ethanol or alcohol induce directly those alterations, or if there are other factors that contribute to this process. So we were looking for a model, as I said earlier. But why didn't we choose the rodent models? Because rodent models, of course, they are widely used in research, but in the set of alcohol use disorder, they present a lot of limitations. So rodents, they have natural aversion for alcohol, so the administration route is not the same as in humans. They also metabolize 5 times faster alcohol than us, so it's really difficult to reach higher ethanol concentration in their guts. They also present a lot of differences in their immune system and the microbiota. And after all, it's quite impossible to reproduce advanced liver disease with this model, even with higher ethanol concentrations. So based on those limitations, we were looking for a model as close as possible to the in vivo condition. And that model that we chose is the intestinal organoids, also called Enteroids. So what are organoids? So organoids are a tiny version of organ that we culture in vitro. They can be derived from different organs and from different parts of the body. But as you might already know, I'm going to talk about the intestinal organoids. So how do we generate these enteroids? So first we collect biopsies from patients in the clinic, and then we collect those biopsies in PBS. Then later we incubate them in EDTA, And with a mechanical shaking, we can isolate the crypt from the biopsy. Then we put them in culture to generate our organoids. So usually they grow from day 0 to day 6, having this round shape. And then from the 6, they start their differentiation. And at the 14, you have really fully differentiated organoid with crypts and villi architecture as in vivo. In our lab, we're using 2 different techniques of culture. First, we are using a 3D. So the thing is, in the 3D, the organoids are cultured in matrigel. So matrigel represents here an extracellular matrix that mimics the in vivo microenvironment. We also use a 2D culture. So in the 2D, with the 2D culture, we culture the organoids on a matrigel layer, and then in the transworlds. So one of the advantage of this model is it allows us to perform a co-culture with immune cells that we are doing right now in the lab. And also in 2D, you have the apical phase that is in contact with the cell culture medium, so mimicking what we have in vivo. So on this slide, you have the images of what we really have under our microscope. So on the top, you have the organoids. Here at the 3, you can see the round shape of the organoids. Then you can see here at the 7, we start losing this round shape because they start their differentiation. And at the 14, you have fully differentiated organoids with crypts and villi architecture. And at the bottom with the 2D culture, you can see that the morphology is a bit different. Yeah, we still have at the beginning the round shape, and then at the 10 you have a differentiated enteroids. But at the 14 we don't have isolated organoids anymore. We have organoids that's open and attached to each other, so you have a single monolayer of cells. So we were asking, we wanted to know if this model is really representative of the gut epithelium and if we have the different epithelial cells in this model. So we first retrieved the organoid from the matrigel, then we digest them in single cell before we incubate them with the antibody. So in this case, we use a LGR5 marker to detect the stem cells. We also use a MUC2 to detect the goblet cells, the lysozyme for the Paneth cells, and the CHDA for enteroendocrine cells. So as you can see with these 2 figures here, we have the presence of all the population. You can see that we have the population of the stem cells that represent here 3.65%. We also have the Paneth cells that represent 2%, and we have a lot of MUC2 positive cells and enteroendocrine cells. So based on these results, we can say that we have all the epithelial cells that are represented in the enteroid model. To confirm the results of the previous slides, we performed an immunostaining and we used confocal for the imaging. So first, we identify and localize ZO1 protein because this protein is really important for us because it's involved in the maintenance of the membrane integrity. So as you can see, ZO1 protein here, we can see its presence between the epithelial cells. We also use OLFM4 marker, which is a marker of stem cells, but also the cells that are not fully differentiated yet. So we can see that in our organoid model, we have the presence of OLFM4 marker here in red. On top of that, we also checked the presence of MUC2, the goblet cells, by using MUC2, as I mentioned earlier. So you can see here inside the organoid, the presence of MUC2 positive cells. And this is really represented here in 3D, where you can see clearly the presence of these cells inside the organoid. Then we wanted, since if you remember at the beginning of the presentation, I talked about an increased cell proliferation in AUD patients. So we also checked the proliferating cells with KIC7. So you can see in green here that we have, we can detect the proliferating cells in the organoids. The last thing we checked is if the organoid could produce mucus. So in this case, we use a whey-germ agglutinin, which is a lectin that binds to glycosylated protein. So its positivity here means that we have mucus production inside these organoids. So since we are fit with the technology, the next question we ask, what are the differences between control and AUD organoids? So here on the top left corner, you can see the control organoid, and on the right, you can see the AUD organoid. So as shown, we can see that there are differences between AUD and control organoids. Some of AUD enteroids do not have a clear defined crypt and villi architecture, but some of them are highly structured. So based on those results, we can say that the heterogeneity between the patients are also kept in the culture, which is a really good thing. So on the top of that, we quantify the gene expression of the same epithelial cells. As you can see here on the left, we have a MYC2 gene overexpression in AUD compared to the control. But in contrary, we have the downregulation of LGR5 for the stem cells gene, and also the lysozyme that is for the Paneth cells. And these results are confirmed by the FACS result here on the right, where we can see that the most abundant cells are the MYC2, the goblet cells. So we can say based on those results that the results that we have here are the same that with the ones that we obtained previously on the patient biopsy. So as you know, we are working on alcohol use disorder. So in this case, we incubated the organoid with ethanol to see what are the effects of the ethanol in these enteroids, and if we will have the same effects as in vivo. So we incubated the enteroid with ethanol for 5 days. And then we performed immunostaining as previously, and we used a KIC7 marker. And then the quantification of KIC7 positive cells per nucleus showed that we have an increased cell proliferation with rising ethanol concentrations. So that confirms the previous result that we obtained on those patient biopsy. Finally, the last thing we checked is how harmful is ethanol to the organoids. So we performed same experiments as previously, but in this case, we incubated the organoid with ethanol for 9 days. So meaning 2 days, the second day after passage until they achieve their full maturation. And in this case, we use propydium iodide that stains the dead cells, but also gives information on the membrane integrity. So that means the propydium iodide cannot enter the organoids if the membrane is still intact. So as you can see with these confocal images, you can see that with a higher ethanol concentration, we have a higher cell death stained here in red. And the quantifications also show that we have more cell death with higher ethanol concentration. So I would like to end this presentation by giving you the advantages, but also the limitations of our model. So as for the advantages, we can say that the organoids are host-derived, and we saw during this presentation that they can keep the phenotype, which is really good. And also it contains all the different type of epithelial cells and offer us the possibility to perform co-culture. But as every model, there are some limitations. So we have, with our model, we have an absence of the microbiota, which is really important. But we can still incubate the enteroids with bacteria-derived products. This model is also a single compartment. So it's single compartment, but if you remember at the beginning of the presentation, I showed you the different layers of compartment. We have the mucus, we have the epithelium, then we have the lamina propria. But to overcome this limitation, we can use the co-culture model with the immune cells. And one of the biggest disadvantage also is the loss of phenotype after several passages. So that's why we work with organoids that do not have a higher number of passage. So to conclude, we can say that the model is representative of the gut epithelium, and also it's reproduced the phenotype, so we can study each patient precisely. And for the future perspectives, we want to perform multiplex on the organoid to identify and localize the different cell types, as we performed here on the duodenal biopsy. We also started collecting samples to perform single-cell RNA. And after all, we want to assess the different pathways that are altered in those patients. And finally, we want to modulate the model to try to correct the different alterations that we saw with this organoid. So this is the end of my presentation. If you have any question, I will be more than happy to answer them. I would like to thank everyone, and especially Grant team. Thank you.
Thank you, Amy, for the nice talk. But before we go to the question of the chat, I wanted to ask one question myself.
Yeah.
So you said that you were using that the organoids were taken first from biopsy, were based from biopsy from patient, and I was wondering from which part of the intestine it was taken, and if you can see that it has an influence if you take other parts of the intestine.
Okay, thank you, Maud, for your question. Sorry, I didn't mention it or during the presentation, but we used the duodenal biopsy organoids.
Yeah.
And yeah, of course, the part of the intestine that we use to generate the organoid has an impact, because for some parts of the gut, like if we take the duodenum, the cell composition is different from if we take maybe ileum or other parts of the intestine.
Okay. Thank you. I will now go to the question from the chat. So the first one, are alcohol use disorder organoids replatable for many passages considering reduction in stem cells?
For now, we compared organoids that went to several passages, and we can see the difference in the proportion of the stem cells. Even in culture, when we put them in culture, we compared like day 1 and day 14 when they are fully differentiated. So at the 14, we don't have any more stem cells. So also for the passages, as you asked, we have the reduction of stem cells depending on the number of passages.
All right.
I don't know if I'm clear enough.
I suppose. So the next question is, integrity of the intestinal mucosal barrier is key. To maintain and repair this mucosal barrier integrity in vivo, the presence of intact enteric glia is crucial, but these are clearly lacking in the proposed organoid model. to what extent might this lack of enteric glia have an impact on the translational validity and value of your organoid model?
Please, can you repeat the question?
Yes. So there is a clear lack of the intact enteric glia in your model, and the person is wondering if this can have an impact, to which extent this will have an impact on the translational value of the organoid model.
Okay, thanks for the question. So as I already said during the presentation, this is a single compartment model, so we don't have all the different compartments. So yeah, of course, as every model, it has limitation, but I don't know quite precisely about the glia, but I know for instance, for the immune cells, we have the communication between the epithelial cells and the immune cells. So that's why we are proposing the co-culture to overcome this limitation. But we cannot also, because it's really complex 3D models, so we cannot have every cells that will be present, every compartment, but we can get close to that. That's why we can have two compartments, and also maybe for the bacterial part, we can incubate with the bacteria derived products, but yeah, we can still have limitations.
Okay, thank you. The next question is, why do you use Matrigel? Do you think it is the best scaffold, or do you think there are better alternatives?
Okay, for the Matrigel, we use it because it's well known. It's well known its use in the literature, but also when we tested this Matrigel with the organoids, the growth is really optimal. But there are also others such as collagen, or we also have hydrogels that do not contain animal because the material is coming from animals. So that's why some of people also use hydrogels. But I do not know at what extent the impacts on the culture, because since we started, we just using the material.
Okay. So I don't see any other questions in the chat. So we can now end this webinar. But before we end, I would like to thank very much the two speakers of today for their nice presentation. Thank you as well for the people to the people who attended this webinar. I would like to remind you, if you would like a certificate of attendance, you can put your email address in the chat, or you can also contact me later on. If you have any suggestion for other topics, or if you would like to present your work during the following webinars, you can also indicate it in the survey when you close this webinar. And the recording will be also posted in the coming weeks on the YouTube channel. So thank you very much to everyone, and I hope to see you soon. Bye.